Welcome to this tutorial on acid-base balance, brought to you by Medical Biochemistry Power Capsule series. This video covers the following CBME curriculum topic: describe the processes involved in maintenance of normal pH, water and electrolyte balance of body fluids, and the derangements associated with these. Now, for recapitulation of your knowledge. An acid is any hydrogen-containing substance that is capable of donating a proton to another substance. Proton is basically a hydrogen ion. A base is a molecule or ion able to accept a proton from an acid. Therefore, acids are proton donors, bases are proton acceptors. pH can be considered as abbreviation for hydrogen potential. It is a numerical figure expressing the acidity or alkalinity of a solution on a logarithmic scale on which 7 is neutral pH. Lower values are more acid and higher values are more alkaline. pH is an indirect indicator of the ratio of hydrogen ions and hydroxide ions. Alkalis are specifically water-soluble bases. All alkalis are bases, but all bases are not alkalis. Alkalis yield hydroxide ions when dissolved. Normal metabolic processes result in the production of large amounts of carbonic acid and lesser amounts of sulfuric, phosphoric, and other acids. During a 24-hour period, a person weighing 70 kilograms respires approximately 20,000 millimoles of carbon dioxide, the volatile form of carbonic acid, through the lungs, and excretes about 70 to 100 millimoles of non-volatile acids through the kidneys. Carbon dioxide and acids are transported to the lungs and kidneys via the extracellular fluid and blood with no appreciable change in their pH. pH of arterial blood is within 7.35 to 7.45, and pH of venous blood is within 7.32 to 7.38, hence, blood is mildly alkaline. The pH of the body has to be maintained within narrow normal limits. Any deviation is not conducive to life. Abnormalities of acid-base balance, frequently accompanied by abnormalities of water electrolyte balance, is a major reason for morbidity and mortality. Normal metabolism produces carbon dioxide, lactic acid, keto acids, etc. Imagine what would happen if this huge amount of carbonic acid and the other non-volatile organic acids keep on accumulating. Survival wouldn't be possible beyond few hours. What can happen if pH becomes high or low? Everything goes into disarray, and chaos breaks out. Various molecules in our bodies, such as proteins and nucleic acids, have ionic character because they have net electrical charge. These molecules, in order to maintain their structure and function, need to have electric charge or ionic character. However, the ionic character of these molecules depend on the ionic character of the surrounding fluid. Change in body pH changes the ionic character of body fluids, thereby causing a change in the ionization status of these molecules. This adversely affects their structure and function. When pH of body fluids deviates from the normal range, molecules such as proteins, nucleic acids, etc., change their ionization status. Consequently, their intramolecular configuration changes, which affects their normal functionality. It's because function depends on their structure and configuration. This flowchart provides a simple description of the events. pH of body fluid changes, ionic character of body fluid changes. Ionization status of charged molecules, such as proteins and nucleic acids, is altered. Structure and functionality of charged biomolecules is affected. The pH status of our body is a reflection of the constancy of the concentration of hydrogen ions and the concentration of various negatively charged basic ions. Hence, body pH is all about the relative concentration of hydrogen ions and negative basic ions, such as hydroxide ions. This body pH is kept within a tight limit under normal circumstances. In order to understand acid-base disorder, we first need to understand how normal acid-base balance is maintained. As said earlier, various types of acids are continuously produced in the body, bases may also get into the body, hence, acid-base balance is all about keeping their relative concentrations stable and within the acceptable range. So, what does the body do to maintain normal pH balance? Obviously, the body has to eliminate the acids. Elimination of carbon dioxide and protons is carried out through lungs and kidneys, respectively. So elimination is the way to keep the acid level in check, but only elimination mechanism would not be sufficient, and we need to have other mechanisms in place. Maintaining constant pH is made possible by the combined and coordinated action of body buffers, and respiratory and renal regulatory mechanisms. Elimination is the final step. It is the final line of defense. Before that, there is a first line of defense. This first line of defense is needed for immediate neutralization of acids. That's important because the lungs and kidneys need some time to eliminate acids. Hence, a quick-acting mechanism is required as a preliminary step. Also, the mechanism of elimination of acids by lungs and kidneys requires a pre-processing of the acids through neutralization. So, we have first line of defense and the final line of defense. This immediate neutralization and pre-processing is achieved through the action of buffer systems. So, we have two sets of mechanisms for acid-base regulation: the short-term buffer system, which is the first line of defense, and the long-term regulatory mechanism involving lungs and kidneys and which is the final line of defense. These two systems work cooperatively to achieve the purpose. Imbalance occurs whenever these systems get overwhelmed and are unable to handle the load of acid or alkali. From your knowledge of college-level chemistry, you know what buffer systems are. A buffer system is a chemical system that resists abrupt changes in its pH. This means that, whenever some acid gets into a buffer system, the pH of the system doesn't get lowered. Similarly, whenever some base gets into a buffer system, its pH doesn't rise. The buffer system has its own pH, and it remains constant even after addition of acid or base. Of course, every buffer system has its own capacity, and the ability to resist changes is not unlimited. Buffer systems or buffer solutions are such that their intrinsic pH remains constant despite addition of acids or bases. What are the constituents of any buffer system? A buffer system is a mixture of weak acid and its salt. For example, a mixture of acetic acid and its salt, sodium acetate, comprises a well-known buffer system, which is the acetic acid buffer system. Together, they are known as conjugate acid-base pair. Acetic acid and acetate anion comprises a conjugate acid-base pair. Acetic acid is a weak acid. This means that molecules of acetic acid do not dissociate fully in solution. At any given point of time, only a fraction of acetic acid will remain dissociated as acetate anions and hydrogen ions. For example, if there are 100 molecules of acetic acid, only 10 may remain dissociated and 90 will not be dissociated. This means that only 10 hydrogen ions will confer acid character to acetic acid solution. In contrast, in case of strong acids, such as hydrochloric acid or sulfuric acid, out of 100 molecules, all 100 would be dissociated into protons and chloride or sulfate anions, as the case may be. More protons means more acid character. Remember, only weak acids can form buffer systems. Strong acids can never form buffer systems. In a buffer solution comprising of acetic acid and sodium acetate salt, the various ions present are as follows. Acetate ions contributed by acetic acid and sodium acetate. Sodium ions contributed by sodium acetate. Hydrogen ions contributed by acetic acid as well as water. Hydroxide ions contributed by water. It's important to understand that water also ionizes, though very slightly, because water is weakly dissociable. It's also important to understand that whatever acid character this buffer solution possesses is due to those hydrogen ions. But because the concentration of hydrogen ions is less, acidity is also less. Fact of the matter is that This low acidity can also be kept at a constant pH level even after addition of acids from external sources. The hydrogen ion concentration of a mixture of acetic acid and sodium acetate doesn't change. Hence, it's a buffer mixture. But how is that possible? What happens is as follows. Suppose hydrochloric acid is added to this acetic acid buffer. It dissociates completely into hydrogen and chloride ions. The hydrogen ions immediately get gobbled up by acetate ions present in the buffer solution because, unlike chloride, acetate is a very strong base and an attractor of hydrogen ions. Thereafter, we get more undissociated acetic acid molecules. The extra hydrogen ions coming from hydrochloric acid is unable to raise the acidity of the solution because they get combined with acetate ions. We need free hydrogen ions for acidity. Hydrogen combined with something else cannot confer acid character. Contrarily, if hydrochloric acid is added to plain water instead of buffer, then the liberated hydrogen ions cannot combine with anything because there is nothing to gobble them. Hence, free hydrogen ions in plain water raises acidity. Pause and go through the reaction process depicted here to understand what actually happens. In this acetic acid sodium acetate buffer, the supply of acetate ions comes from sodium acetate. This sodium acetate salt serves as the reservoir of acetate ions. As long as the quantity of salt in the buffer is sufficient, any added hydrogen ion from external source will keep on combining with the acetate anion. On the other hand, added hydroxide ions, in the form of a strong base, combine with hydrogen ions derived from acetic acid and form the weakly dissociable water molecules. Hence, pH does not become as alkaline as would happen in absence of the buffer. Hydroxide ions can be buffered as long as some acetic acid remains to supply the hydrogen ions. Sodium acetate is the reservoir of acetate ions for buffering against hydrogen ions. Undissociated acetic acid is the reservoir of hydrogen ions for buffering against hydroxide ions. It's also important to remember that buffer systems to have an intrinsic pH owing to their weak dissociation capability. They are not neutral, but, whatever be their pH value, they can maintain it constantly despite addition of hydrogen ions or hydroxide ions from external sources. The takeaway from the discussion so far is that, in order to protect any system, whether living system or non-living system, from severe pH fluctuations, buffers can play an important role. Their mode of action is also immediate. The ionic dynamics within a buffer system or buffer solution is such that they can resist changes in pH, which means that they can keep the relative proportion of hydrogen and hydroxide ions constant. The body has major and minor buffer systems. We don't have the acetic acid buffer system in our body, rather, our buffers are something else. Needless to say, mechanism of action is similar in principle to that of acetic acid buffer, only the chemical nature of the components differ. The bicarbonate buffer system is the major one and its site of action is extracellular. Others are mainly intracellular, though protein buffer has significant extracellular action too. The major buffer systems operating within our bodies include carbonic acid buffer system or bicarbonate buffer system, phosphate buffer system, protein buffer system, and hemoglobin buffer system. Please pause for a couple of minutes and go through this table depicted here. Note the conjugate acid-base pairs and their components. Carbonic acid and sodium bicarbonate are the components of the major body buffer system, known as bicarbonate buffer system. Protein molecules, being negatively charged, can also quench hydrogen ions, and so can form buffers. Hemoglobin can act as buffer because when pH becomes acidic, it can bind to excess protons. Hemoglobin releases protons when pH becomes alkaline. In fact, hemoglobin can bind with both carbon dioxide and protons. When there is excess of carbon dioxide in blood, carbon dioxide reacts with amino groups of hemoglobin to form carbaminohemoglobin. Also note that deoxygenated hemoglobin and oxygenated hemoglobin forms another buffer pair known as the ABC hemoglobin buffer system. Hemoglobin is therefore another versatile blood buffer system. Each of these four buffer systems has a tightly controlled ratio of their acid and salt component. The intrinsic pH of these buffers depends on this ratio, as evident from the Henderson-Hasselbalch equation. It's obvious that pH of a buffer solution depends on the concentration of the acid and base components and their relative ratio. Changing this ratio can change the pH of the buffer. This is the Henderson-Hasselbalch equation. pH is equal to pKa plus logarithm of the ratio of base by acid. Here, pKa is a constant, known technically as acid dissociation constant. Base within brackets is concentration of base. Acid within brackets is concentration of acid. This equation is the fundamental equation that governs and explains the ionic behavior of acids and bases in solutions. At the cost of repetition, I am again stating that this equation clearly shows, mathematically, that pH depends on the ratio of base and acid components. The pKa has a constant value for a particular acid-base conjugate pair, but is different for different pairs. This pKa is dependent on the strength and chemical nature of acids and bases. Our body needs to maintain constant pH, hence, the buffers need to have definite ratios, which would maintain their pH at physiological range. You need to understand that our entire body fluids are practically buffers because buffers suffuse our body fluids. The blood is a buffer solution because it has bicarbonate buffer and hemoglobin buffer. Intracellular fluid is a buffer because there's buffer present within cells. Our body fluids are physically inseparable from the buffers they contain, hence, when buffers maintain their pH, ultimately the body pH gets maintained. And if the buffer pH changes, the pH of body fluids, and the body as a whole changes. For any particular buffer system, this ratio is the determinant. The buffer system tries to maintain this ratio despite addition of acid or base from external source. And because it's a ratio, the relative proportions of the components are more important than the absolute quantities. As an example, if the acid component, which is denominator, increases, the base component too should increase if the value of the ratio has to be kept constant, and vice versa. It's also evident from the equation that, if the base divided by acid factor in the equation changes, the value of pH will also change. Let's try to understand what actually happens. Suppose the body starts producing excessive lactic acid. This leads to a rise in hydrogen ions. These hydrogen ions will be buffered by the salt component of bicarbonate buffer, that is, sodium bicarbonate, to yield more carbonic acid. As a result, the salt component decreases and acid component increases. That leads to a lower value for the factor, base divided by acid. The logarithm value therefore decreases, and thereby the pH value decreases. This means that acidity increases. But in reality, the pH remains constant. This happens in two ways. Firstly, the body synthesizes more sodium bicarbonate to constantly replenish the used-up sodium bicarbonate. The concentration of carbonic acid gets lowered by elimination of carbon dioxide through lungs. Therefore, the net result is that a strong acid, like lactic acid, gets neutralized and the weak carbonic acid is formed. But this carbonic acid cannot decrease the pH because it is continuously eliminated through the lungs as carbon dioxide. Please recall that carbonic acid and carbon dioxide are in reversible equilibrium and can easily interchange, depending on their concentration and partial pressure. Fact of the matter is that lactic acid is prevented from decreasing the body pH and cause any damage, and mathematically speaking, the base-by-acid ratio is therefore maintained by synthesizing more sodium bicarbonate and by eliminating carbon dioxide, which can be considered as the gaseous form of carbonic acid. The Henderson-Hasselbalch equation ensures that the pH is not changed. The carbon dioxide from carbonic acid is eliminated through the lungs. Hydrogen ions get incorporated into water molecules. As already mentioned, dissolved carbon dioxide, being in equilibrium with gaseous carbon dioxide, can be easily eliminated because partial pressure of carbon dioxide in alveolar spaces is much less than compared to partial pressure in blood. Please pause for a few seconds and go through these equations. Hydrogen ions combine with bicarbonate to form carbonic acid. Carbonic acid dissociates into carbon dioxide and water. This carbon dioxide remains dissolved for the time being. Upon reaching the lungs, carbon dioxide can no longer remain in solution because of comparatively higher partial pressure in blood. It comes out as gas in the alveolar spaces and gets out through exhaled air. There's an interesting point to be noted here. Buffering by bicarbonate buffer system is closely linked with the RBC hemoglobin buffer system. The bicarbonate buffer system works in conjunction with RBC hemoglobin buffer system. This also involves carbon dioxide transport and carbon dioxide elimination. Hence, bicarbonate buffer plus RBC hemoglobin buffer serves both as an effective first line of defense against acid-base changes, as well as a transportation-cum-elimination system for carbon dioxide, which is a metabolic waste product. The RBC hemoglobin buffer system works synergistically with bicarbonate buffer system. In fact, the bicarbonate buffer system could not have achieved its purpose had there been no RBC hemoglobin buffer. It's an obligatory relationship between these two systems. Carbon dioxide is generated in tissues. Tissue partial pressure of carbon dioxide is high. Carbon dioxide easily diffuses into capillaries and enters venous blood. Carbon dioxide combines with water and aided by the enzyme carbonic anhydrase, found inside red blood cells, forms carbonic acid. This carbonic acid then dissociates into hydrogen ions and bicarbonate ions. This reaction occurs rapidly and spontaneously. You will notice that due to these two sequential reactions, hydrogen ions and bicarbonate ions are ultimately generated within red blood cells. The hydrogen ions are immediately caught by deoxygenated hemoglobin and thus effectively buffered. Hydrogen ions are no longer in free state. Deoxygenated hemoglobin can easily combine with hydrogen ions. So, we now have hemoglobin combined with protons, in short HHB, inside red blood cells. Meanwhile, the bicarbonate ions pass from red blood cells to plasma in exchange for chloride ions that enter into red blood cells. This is known as chloride shift or Hamburger phenomenon. This is an important method of replenishing valuable bicarbonate ions in plasma. The illustration shown here explains the process. From this mechanism, we see that carbon dioxide, which is a metabolic waste product generated in tissues, is ultimately converted into bicarbonate ions and transported. Upon reaching the lungs, where HHB takes up oxygen due to high partial pressure of oxygen, hydrogen ions are released. That's because oxygenated hemoglobin cannot combine with hydrogen ions easily. These hydrogen ions then combine with bicarbonate to form carbonic acid. The carbonic acid dissociates into water and carbon dioxide. The carbon dioxide is then exhaled out. This basically explains the opposite processes that occur in tissues and in lungs sequentially. Bicarbonate buffer system and hemoglobin buffer system are two different systems. Their purpose is also different. Purpose of bicarbonate buffer system is to neutralize strong acids such as lactic acid, keto acid, etc. Purpose of hemoglobin buffer system is to synthesize bicarbonate ions and also to transport carbon dioxide to lungs for elimination. But they work in close cooperation and are functionally linked. Please do not confuse between RBS hemoglobin buffer system and the bicarbonate buffer system. They are different with different roles, but are closely linked. So, what does the synergistic activity of bicarbonate buffer system and RBC hemoglobin buffer system achieve? This combined system takes in carbon dioxide and forms bicarbonate ions. The hydrogen ions formed due to carbon dioxide input is neutralized by RBC hemoglobin buffer intracellularly. The hydrogen ions from other acid sources can also be neutralized by bicarbonate buffer extracellularly. Hydrogen is finally incorporated into water. This system transports carbon dioxide to lungs as bicarbonate. It also transports carbon dioxide as carbonic acid. Therefore, carbon dioxide, which is a potential volatile acid, gets neutralized, transported, and finally eliminated as an useful parallel process. A major buffer system gets constructed, which is the bicarbonate buffer, and which serves as an effective extracellular blood buffer against non-volatile acids. Volatile acid ultimately neutralizes non-volatile acids. Carbon dioxide that gets eliminated through lungs comes from two sources. It comes from carbonic acid that is generated when strong acids are neutralized, and it also comes from carbonic acid generated within red blood cells. The bicarbonate buffer system is unique. It's different from the other three buffer systems. The acid component of the bicarbonate buffer system is composed of a waste product, which is carbon dioxide. Even the base component, bicarbonate, indirectly comes from carbon dioxide. Please recall that carbon dioxide is the largest fraction of acid generated by the body. So nature, very intelligently, utilizes this acid-cum-waste product to do some good work before it gets eliminated. It is used to constitute the body's largest and most significant buffer system. The respiratory center has an important role to play. Participation of the respiratory mechanism in the regulation of acid-base balance depends upon the sensitivity of the respiratory center in medulla oblongata to very slight changes in pH and carbon dioxide partial pressure. It also depends on the ready diffusibility of carbon dioxide from the blood across the pulmonary alveolar membrane into the alveolar air. Hence, the lungs should be healthy so that diffusion of carbon dioxide can take place properly. Increase in partial pressure of carbon dioxide in blood results in increase in pulmonary ventilation due to stimulation of respiratory center. Slight increases in hydrogen ion concentration of the blood can also stimulate the respiratory center. The purpose of this entire mechanism is to ensure that carbon dioxide doesn't accumulate in the body, because if carbon dioxide accumulates, the pH of the body will become acidic, and a lot actually depends on how efficiently carbon dioxide gets eliminated, because carbon dioxide is the product formed after other strong acids have been neutralized. Inefficient elimination of carbon dioxide will hamper the entire set of downstream processes connected with acid neutralization. The excess carbon dioxide is thereby promptly removed from the ECF in the expired air. Conversely, a decrease in carbon dioxide partial pressure in blood or a decrease in hydrogen ion concentration, in case of alkalosis, causes depression of respiratory center, with consequent slow and shallow respiration. This results in retention of carbon dioxide in the blood until the normal partial pressure of carbon dioxide and pH get restored. This respiratory mechanism, therefore, tends to maintain the normal bicarbonate-carbonic acid ratio in the extracellular fluids. The respiratory mechanism determines how much carbon dioxide is eliminated or retained. Please note that only eliminating carbon dioxide through lungs doesn't complete the story. A net elimination of hydrogen ions is also required. This net elimination of hydrogen ions occurs through kidneys, but because it takes time, the body needs immediate defense against hydrogen ions liberated from various acids. It does so by allowing bicarbonate to combine with hydrogen ions and form the less harmful carbonic acid. Carbon dioxide gets breathed out because it's in a loose reversible equilibrium with carbonic acid. The hydrogen ions are carried to kidneys in harmless form as water to be eliminated as such. A net elimination of hydrogen ions from the body is ultimately achieved through the kidneys. The final solution is through the kidneys because that's the major way through which protons can be eliminated. Renal mechanism for maintenance of acid-base balance occupies an important position in homeostasis. Renal mechanism involves mechanisms for bicarbonate ions reabsorption, buffering by phosphate and buffering by ammonia. Let's go through the renal mechanism involved in maintaining acid-base homeostasis. Please observe the schematic diagram. Bicarbonate ions and sodium ions are filtered by the glomerulus and enter the filtrate in renal tubules. Hydrogen ions enter the renal tubules from the renal tubular cells. The hydrogen ions are derived from carbonic acid, which is formed due to the action of carbonic anhydrase. Carbon dioxide inside renal tubular cells reacts with water in the presence of carbonic anhydrase. These water molecules originate in the body and can be considered to contain hydrogen ions that are produced during buffer action. As such, the hydrogen ions that enter the renal tubule owe their origin to acids elsewhere in the body. In the tubular fluid, the hydrogen ions now combine with the filtered bicarbonate ions. This ultimately results in the formation of water and carbon dioxide. The carbon dioxide is reabsorbed into the renal tubular cell. Interestingly, you will notice that, for every bicarbonate ion filtered, another bicarbonate ion is generated within renal tubular cell, which then gets absorbed. Hence, bicarbonate is not lost, but is rather reclaimed. And hydrogen ions are eliminated out by getting incorporated into water molecules. This process ensures that a net elimination of protons from acids can take place and valuable bicarbonate can be reclaimed. In another process, there is net synthesis of bicarbonate ions. There should not be any confusion here. The process described previously pertained to bicarbonate reclamation. This here pertains to net synthesis. Here, hydrogen ions within tubular filtrate combines with a filtered anion such as lactate, sulfate, acetoacid, etc., and is excreted as acid. You will notice that there is synthesis of bicarbonate going on in renal tubular cells and, which is absorbed into the system, but there is no elimination of bicarbonate in the urine. It's because the hydrogen ions, in this case, combine not with bicarbonate, but with other negative ions. End result is that protons are eliminated and bicarbonate is synthesized. We already know that there is net synthesis of bicarbonate within red blood cells. Now we know that bicarbonate is also synthesized in renal tubular cells. Bicarbonate buffering in the body fluids is not the same as bicarbonate buffering in the kidneys. They have different purposes. In body fluids, purpose of bicarbonate buffering is immediate neutralization of strong acids so that they do not cause harm, and to form carbonic acid, which can ultimately be expelled as carbon dioxide. In the kidneys, the purpose of bicarbonate buffering is to eliminate hydrogen ions while at the same time conserving itself by getting reabsorbed elsewhere in the body. Bicarbonate neutralizes strong acids and maintains pH balance. In the kidneys, bicarbonate captures hydrogen ions and eliminates them as water molecules. The complete neutralization of lactic acid can therefore be summarized as shown below. When lactic acid increases, hydrogen ion concentration increases. Hydrogen ions get captured by bicarbonate component of the bicarbonate buffer system. As a result of the reaction between bicarbonate and hydrogen ions, the concentration of carbonic acid increases. Carbonic acid now contains hydrogen from lactic acid. Carbon dioxide gets eliminated through lungs because carbonic acid ultimately yields carbon dioxide. But still, the burden of excess hydrogen ion is still there in the body. These hydrogen ions are now part of water molecules. Finally, hydrogen ions are eliminated by kidneys by a complex mechanism involving bicarbonate mechanism, phosphate mechanism, and ammonia mechanism. The last two will be described subsequently, and therefore, lactic acid is finally made ineffective. Buffering by phosphate is fairly straightforward. Phosphate is considered as the most important renal buffer. In this process, net synthesis of bicarbonate also occurs. Basic phosphate and acid phosphate forms the conjugate acid-base pair. Everything else remains same as it is in case of renal bicarbonate buffering, except the fact that here, hydrogen ions combine with basic phosphate and get eliminated as acid phosphate in the urine. It is also worth noting that hydrogen ions enter into the tubular fluid in exchange for sodium ions by utilizing the common transport protein known as sodium hydrogen exchanger or NHE. Ammonia can also act as a urinary buffer, and is formed by the deamination of glutamine in renal tubular cells by the action of the enzyme glutaminase. Ammonia readily diffuses across the cell membrane into the tubular lumen, where it combines with hydrogen ions to form ammonium ions. Ammonium ions do not pass across cell membranes, so passive reabsorption is prevented. Glutaminase is induced in chronic acidosis, stimulating increased ammonia production and therefore increased hydrogen ion excretion in the form of ammonium ions. The ammonia-ammonium buffer pair becomes active only after the buffering capacity of phosphate is exhausted. Ammonia is an important urinary buffer produced by deamination of glutamine in renal tubular cell. Glutaminase, present in tubular cells, is responsible for this conversion. Its activity is induced in chronic acidosis, thus permitting increased ammonia production. Hydrogen ions produced in the glutaminase reaction probably go to gluconeogenesis pathway along with glutamate. Through this mechanism also, there is elimination of protons from the body and synthesis of bicarbonate. You may choose to pause here for a few seconds and go through the schematic diagram in order to understand the process. Neutralization of bases by the bicarbonate buffer system is comparatively simple. The hydroxide ions of any alkali reacts with the carbonic acid component of this buffer system to yield water and bicarbonate. If the input alkali is sodium hydroxide, then the base formed will be sodium bicarbonate. Hence, addition of any base increases the alkali reserve of the body by increasing the salt component of the bicarbonate buffer system. Final regulation will be done by kidneys. That's because excess sodium bicarbonate cannot be allowed to accumulate, just as excess hydrogen ions are not allowed to accumulate. In alkalosis, breathing slows in order to conserve carbon dioxide. Bicarbonate reclamation in kidneys is minimized. Phosphate buffer is the major intracellular buffer, where it is very effective. It is also a major urinary buffer, about which we have already discussed. It consists of the following components as shown. Buffering is simple and involves the interchange between the acid and base components. Proteins can act as buffers because many of them have amino acids, which behave like weak acids. For example, histidine acts as a buffer because of the presence of imidazole group. The intracellular fluid proteins serve as major buffers. Since ICF volume is around 60% of the total body fluid volume, proteins may be considered as most abundant buffers in the body. However, the interstitial fluid, which forms more than 70% of the extracellular fluid, lacks proteins, and bicarbonate buffer plays a major role there. Protein buffers can be represented as comprising of sodium proteinate salt and HPR acid pair. Principle of working is same as in other buffers described previously. In acidic medium, proteins act as a base. Amino group takes up hydrogen ions from the medium, forming ammonia ions. Proteins become positively charged and hydrogen is neutralized in the process. In alkaline medium, proteins act as an acid. Acidic carboxyl group dissociates and gives hydrogen ion. Hydrogen ion then combines with hydroxide ion to produce a molecule of water. Proteins become negatively charged and hydroxide gets neutralized. The generalized reactions involved have been depicted here for your convenience. Carbon dioxide, which is a potential acid, can also be buffered by formation of carb-amino compounds with amino groups of protein molecules, thus directly fixing carbon dioxide. This reaction achieves the binding of carbon dioxide without passing through the carbonic acid stage. This has also been mentioned earlier in this video in relation to buffering by hemoglobin. Therefore, there are at least 3 ways in which proteins can act as buffers. The components of these 3 separate mechanisms have been shown here. It's important to understand that, even in normal physiological conditions, acids will be generated. The normal balancing mechanisms, or defense systems, can deal with these acids. Problem may arise in either of the 2 situations: if acids are generated in excess due to some disease process, or if the defense mechanisms are compromised due to some disease process. For example, Excessive generation of ketoacids in diabetic ketoacidosis is a situation of excessive acid generation due to disease. For the second instance, we may cite the example of lung pathology, which may cause so severe damage to lungs that carbon dioxide elimination becomes difficult even when acid production is not high. End result in both the situations is that there will be an accumulation of hydrogen ions in the body leading to acidosis that is uncompensated. Same may be said of alkalosis too. Here, there is a net increase of basic ions due to various reasons. A separate tutorial covers disorders associated with acid-base balance and other applied topics. I would like to suggest that you should definitely watch that video too.
Thanks for watching this video. Our next tutorial in the Medical Biochemistry Power Capsule series is titled, Disorders Associated with Acid-Base Balance. The tutorials presented in the MBPC series are not intended to be a substitute for your textbooks. However, we feel that these tutorials can prove to be very valuable as complementary educational aids and for purpose of rapid revision prior to exams. If you have enjoyed this video, don't forget to hit the like button, subscribe for more exciting content, and share with your friends. And do keep watching our channel to remain ahead in the game.